Hi, I'm Al Powers. I'm a physician scientist at Vanderbilt University, and I want to tell you about the research that we're doing in our lab. The mission of our group is to understand and reverse the pancreatic beta cell and islet abnormalities and dysfunction that are present in type 1 and type 2 diabetes. This slide shows the different organ systems that control our blood glucose, shown in the middle of the slide. On the left is the liver that both produces and stores glucose. On the right are skeletal muscle and fat, where glucose is used. The brain at the top controls both the liver, the skeletal muscle, and the fat. And at the bottom is the conductor of this symphony, the pancreatic islet, that makes both insulin and glucagon and influences both the liver and the muscle. Also, bone and GI tract also make some hormones and products that influence our blood glucose. Importantly for type 1 and type 2 diabetes, the number of beta cells, the number of insulin-producing cells, is a critical determinant of whether or not an individual gets type 1 or type 2 diabetes. So our lab is focused on the pancreatic islet, and that's shown in the sphere in the lower bottom. The islet has about 1,000 cells. It's about 250 microns in diameter. It makes up about 1 or 2% of the pancreas, shown in the upper left. The other part of the pancreas makes exocrine or digestive enzymes. So we're interested in both how the islet fits within the pancreas, and we're also interested in specific cells, shown in the right, where schematically we show a beta cell where glucose comes into the cell and causes insulin to be secreted, shown by the turquoise dots at the right side. Now, the islets are an amazing structure. They're really a mini organ. So I want to show you about how the blood flows in with the islet. Those green in the middle, that's a pancreatic islet in a mouse. That red shows the blood rushing into the islet and the blood exiting the islet. If you think about it, the islet has to both sense our blood glucose and quickly secrete insulin. So being highly vascularized is very important. This schematic shows an islet within the dotted lines, and we know that there are more blood vessels inside those dotted lines than there are surrounding the islet in the pancreatic exocrine tissue. We know that the islet gets 8 to 20 times more blood flow. In fact, more than 5 to 15% of the pancreatic blood flow goes to the islet, even though it's only 1 or 2% of the pancreatic mass. The endothelial cells that line those blood vessels are specialized in that they have holes or fenestrations. This leads to a higher partial pressure in the islets than in the surrounding exocrine tissue. Our lab is interested in how this happens. In this slide, I'm going to show you a schematic that we've developed over the years from our studies of showing how the islet becomes vascularized and how it develops its nerve supply. So at the left are islet progenitor cells. They begin to make angiogenic factors like VEGF-A, and that recruits endothelial cells, shown in red, which closely affiliate themselves with the developing endocrine cells, shown in the circles. They then assemble into this sphere of pancreatic islets, and the blood vessels infiltrate those, and one can see the vessels coursing through the islets there. And then at the right is a highly vascularized islet with the blood vessels shown in red and the nerve fibers shown in blue. We know that blood flow begins early on, but it doesn't begin until the islet already has blood vessels and it already has endothelial cells. And then once those endothelial cells assemble into the blood vessels, then the nerve fibers begin to infiltrate, and we see them coming on here, and we know that the blood vessels provide a scaffold for the islet nerve cells to grow inside that. So the model that we've developed and we're working on is how an islet on the right, highly vascularized, highly innervated, how does that happen? We know it makes angiogenic factors that recruits blood vessels and endothelial cells, shown on the left. We know also that endothelial cells give developmental signals or cues to the beta cell. Shown on the lower left is work from the Melton and Zaret labs, where the endothelial cells that line the aorta, shown in red, are in close proximity to developing pancreas, shown in blue. And if you don't have those blood vessels, you don't get a pancreas or a pancreatic islet to develop. So we're interested in how this crosstalk goes back from the islet to the endothelial cells and the endothelial cells back to the islet. We're also interested in how human cells work. So I'm showing you a movie here. On the left is a mouse islet, and on the right is a human islet. Think about this as a ball of cells, and you're going up and down in the z-axis of that cells. One sees that the mouse islet, The insulin-producing cells, which are green, are on the central core. Around the perimeter are the alpha and delta cells that make glucagon and somatostatin. Look how different the morphology and the pattern is in the human islet on the right, where the green insulin-producing cells are dispersed throughout the islet, as are the alpha cells that make glucagon and the delta cells that make somatostatin. So one of the things that's become clear over the last decade that there are fundamental differences in how the mouse islet and the human islet works. And if one stacks up all of those Z sections, one sees the mouse islet on the left and the human islet on the right. This has led to a number of investigators to focus on the human islet, and I show you the Human Islet Research Network, which is funded by the diabetes branch of the NIH. We're part of that, as are a number of laboratories around the country that are studying human islets and how they're involved in type 1 and type 2 diabetes. I want to show you about infrastructure that we've created to study the human pancreas and the human islet. This begins with organ procurement organizations, shown in the gray box at the top, where we communicate with them, and when organs are retrieved for transplantation, if the organ doesn't go for pancreas transplant, it goes to a collaborator, Rita Bettino, that we have in Pittsburgh, and Rita separates the pancreas into the tissue and to the islets. And then we have an interdisciplinary network of investigators that are at Vanderbilt, Stanford, Mount Sinai, UMass, University of Pennsylvania, and Caltech, where we're studying either the tissue or the islets. Let me just show you some of the studies that we've done with these isolated islets. In this, we take human islets, and the syringe shows how we inject them underneath the kidney capsule of a mouse that's immunodeficient that allows these human cells to function in vivo. We then treat the mouse with a variety of different things to induce insulin resistance or to induce obesity, and we look to see how the human islets perform compared to mouse islets. In the panels on the right, we show that there are proliferating cells in the mouse islet, but not in the human islet. So things that stimulate proliferation and growth of mouse islets in this setting don't appear to stimulate human islets to do that. But this model where we can introduce mouse islets into human islets into a mouse allows us to study the islets in vivo. Our lab is also involved in how to quantify the mass of the pancreas and the mass of the islets. And I show you a mouse model that we created where the beta cells, the insulin-producing cells of the mouse, are marked with luciferase, like the firefly that we see glowing in the summertime. And when one gives the animal luciferin, these cells begin to emit a light that we can register. And so we use this light to quantify how many beta cells are in a mouse and how we might increase the number of those beta cells, or why, when diabetes is developed, those beta cells decline. We're also interested in other technology, and we're using things in collaboration with Jack Verosco, shown in the upper left, and the Vanderbilt University Institute for Imaging Sciences, where we're quantifying using MRI and latest analytical techniques the pancreas in individuals who are developing type 1 diabetes. So for example, on the left, this is a cross-section through one of the control individuals, and we're looking at the pancreas, and it's in the middle, about halfway between the top and the bottom, and the left and the right. And then an individual who had just developed type 1 diabetes, we're measuring the pancreas in those individuals and controlling those two groups. The text at the bottom shows that we're studying recent onset type 1 diabetes. The average age so far is about 13 years. We're scanning them with 100 days of diagnosis, and then we're repeating the scan 6 and 12 months later. So far, we and others have found that the entire pancreas in type 1 diabetes appears to be smaller than in the control individuals. Why that is, we don't know, but that's one of the things we're very interested in studying. So our lab at Vanderbilt is interested in how the pancreas functions. We're interested in those islets, in those clusters of cells, and we're also interested in the individual cells as shown schematically on the right. So if you want more information about our lab, the website at the lower left will tell you that. And I want to acknowledge the people in our research group: Christy Amat, who's an MD PhD student; Radhika, who's a research technician; Marcella Bursova and Chunwan Dai are my faculty colleagues in the group. Daniel Dean is a postdoctoral fellow. Rushna is an MD PhD student. Nathaniel's a PhD postdoctoral fellow. Nora Caton is a graduate student in our lab. Neil Phillips, a postdoctoral fellow. Greg Poffenberger, a senior research technician. Diane Sanders, a graduate student. Elaine Shostak, a senior research assistant. And Jack Walker, an MD PhD student. We really have some outstanding collaborators shown in the middle of the slide, and they're at a number of institutions such as Caltech, Mount Sinai, Stanford, and the University of Massachusetts. We've been fortunate to be supported by a number of funding agencies such as the NIH, the JDRF, and the VA. So if you want more information about our lab, please visit us at the website in the lower left. Thank you very much.